Hello, my name is Arman, and today I'm going to be talking to you about the intrinsic immune functions of IFI16 and IFIX. This presentation will cover some of the research that I've done in Dr. Barker's immunology lab here at Drew over around the past 2 years. So to start off with a little bit of background, the immune system has what we like to call layers of defense. We start off with barrier defenses, which are physical structures such as skin that prevent the pathogen from entering the body. Then go on to intrinsic immunity, which are intracellular factors that prevent the pathogen from taking hold of cellular machinery and using it to its advantage. We then move on to innate immunity, which involves that cell sensing that there is a pathogen present and calling other immune cells and factors to aid. And finally, it ends with adaptive immunity, which is a specific response mounted against a pathogen depending on its nature. So to go a little bit more into innate immunity, it relies on the sensing of broad classes of foreign molecules, known as pathogen-associated molecular patterns, or PAMPs. These PAMPs are bound to by our own cellular receptors known as pattern recognition receptors, or PRRs, which, after activating a signal transduction pathway, trigger the transcription of inflammatory cytokines, cell signaling molecules which then go on to alert other immune cells and factors that there is in fact a pathogen present. So intrinsic immunity, on the other hand, is comprised of constitutively expressed intracellular defenses. So what that means, and as this image shows, is that during the life cycle of a pathogen that enters the cell, such as a virus, there are particular proteins that prevent at each stage of the viral life cycle the virus from utilizing the resources and machinery within the cell to produce more of itself. So we're going to be focusing on pattern recognition receptors IFIX and IFI16, a class of proteins known as pi-HIN proteins because they each have a pi-RIN protein interaction domain, shown in gray, and at least one HIN200 nonspecific DNA binding domain, shown in black, and in the case of IFI16, also white. Now, both of these have been shown to be sensors of foreign DNA that trigger the expression of the inflammatory cytokine interferon type 1. So on top of that, however, our IFI16 has been shown to have various intrinsic immune functions, including having been implicated in a variety of immune system processes, autophagy, ubiquitination, cellular stress responses, and the modulation of chromatin, as well as transcription in general. Now, all of these relate to the way the cell responds to viral infection, whether it's through responding to cellular stress, to activation of the immune system, or changing the cell's own gene expression in a way that makes it harder for the virus to utilize cellular machinery. So my questions were to see if IFIX, which shares IFI16's innate immune functions, also shares its intrinsic immune functions. I decided to focus on one particular aspect of IFI16's intrinsic immunity, which states that it inhibits the HIV LTR promoter. So I asked, does IFIX also function to repress HIV-1 transcription through inhibition of the LTR promoter? And I hypothesized that given the similarities between the two proteins, it would. The way we measured the transcription from the LTR was through an experiment known as a luciferase assay. This experiment involves having the LTR control the expression of a gene known as a luciferase, which when exposed to the right reagents catalyzes a reaction that leads to the emittance of photons. By being able to measure this relative emittance of photons, or luminescence, we're able to see relatively how much protein was expressed, and therefore the activity of the LTR. And we found that in the case of both pi-HIN proteins, there was dose-dependent inhibition of the LTR, which means that as we increase the amount of the gene for these proteins that we put in, we saw a relative decrease in the amount of luminescence, therefore decrease in the amount of luciferase, therefore decreased activity of the LTR. So from this, we were able to conclude that IFI16 and IFIX both function similarly as intrinsic immune proteins against HIV-1. We found also that literature which identifies IFIs solely as pattern recognition receptors aren't exactly complete, and there may be more shared functions that these proteins have. I'd like to thank TriBeta and Novo Nordisk for funding these experiments, as well as my mentors Dr. Barker and Dr. Dunaway for their help, and Thomas Potts for his previous research. Attached are my references.